Dear students, today we are going to see about the porins. The objectives are of the today's class is to see the basic concepts of what are the porins, what are the structures, what is the structure of porin, and what are the functions and importance of porins. So what do you mean by porins? Porins are the proteins found on outer membrane of gram-negative bacteria, especially gram-negative bacteria, and these play a crucial role in maintaining structural integrity of bacterial cell and regulate the movements of molecules across outer membrane by selective permeability. And the porins are water-filled channels or cylindrical structures. Uh, these are permeable to hydrophilic substances and first discovered by Hiroshi Nakaido in gram-negative bacilli. Now it has been seen also in a gram-positive bacteria also, but it is having limited variety. So porins are present on the outer membranes. As you can see, the porins, the location of porin, you can see the porins are here, which are located here, which are red colored, inserted on the outer membrane proteins of gram-negative bacilli. Here one another diagram where you can see the porin, the three molecules of the proteins are seen, that is called as homotrimers, which is present on the outer membrane protein. So basically it is made up of beta plated sheet of proteins. So it is formed when two or more polypeptides line up side by side. You can see the polypeptides which are lying side by side, and the individual polypeptide is called as beta strand, and each beta strand is fully extended, and they are stabilized by hydrogen bond or amino and hydroxyl bonds, carbonyl groups of adjacent chains. So this is the structure of porin. You can see beta sheets, then beta strands. Individual is a beta strand and beta sheets, and about 16 to 24 such beta strands are found in the porins, and they are arranged in anti-parallel fashion to form cylindrical structure called as beta barrel structure, and individual strand binds together by loops. So as already I told you, homotrimers, most of the times three molecules are there. They remain together by hydrogen bonding and dipole-dipole interactions, and there is formation of cylindrical structure, which is water-filled structure. So they are also described as beta barrels. The hydrophobic residues of the proteins, porin proteins, are oriented towards the exterior, outside, which will come in contact with outer membrane protein, and the inner part, that is the hydrophilic, they are oriented towards the inner pore. So this is the top view of porins, homotrimers. Three molecules are there, and these structures, the previous slide as well as these structures, have been detected by X-ray crystallography, sequencing protein primary structure. So these are the methods of detection of porins. So what are the functions of porins? Porins are helpful for transport of nutrients and waste, transport of nutrients such as amino acids, sugars, required ions, then waste products are excreted out. It also helps in maintenance of osmotic balance and antibiotic drug resistance, especially as a medical field. Antibiotic drug resistance in gram-negative bacilli, it is predominantly acquired by the modification of porin proteins. The size of porin is which will permit only about 600 to 800 Dalton size of the molecules. However, there are certain antibiotics like daptomycin, vancomycin, which are having 1400 Daltons. So these antibiotics will not penetrate gram-negative bacilli, and therefore they are not effective for gram-negative bacilli. Porins are also helpful for cell signaling and communication. So these are the further readings. I request all the students, PG students, to go through the other articles which are there and have the in-depth analysis of the structure of porins and its functional importance and what are the research says about that. So take home message for this presentation is porins are present on outer membrane proteins, and these are helping in regulation of transport of molecules. The structural and functional role of porins is very important, which will maintain the integrity structure, maintaining the ionic balance in the interior and exterior of the bacteria. And most important is, as the medical field is concerned, drug resistance due to modification of the transport. Most commonly gram-negative bacilli like E. coli and Klebsiella. Thank you for your patient listening. Thank you.